Good evening and welcome, everyone. Thank you for joining us for this really special event and one of my favorites of the year. I'm Dr. David Dodick. I'm the chair of the American Brain Foundation. And as many of you know, the American Brain Foundation funds research across the entire spectrum of brain diseases and disorders, including Alzheimer's and other dementias, Parkinson's disease, multiple sclerosis, stroke, epilepsy, cognitive aging, and many more. Because we know that many brain diseases are interconnected. There's similarities in the biologies of these diseases. So funding research across the full spectrum of neurological diseases is really one of our best hopes for finding improved treatments, preventions, and cures. Brain disease is a global issue, and this year in particular, the global impact of brain diseases has become especially apparent. The Global Burden of Diseases study was recently published, or at least another iteration of it was published just a few months ago. And it estimated that over 1 in 3 people around the world are affected by a neurological condition, meaning that it is now the world's leading cause of ill health and disability worldwide, and the second leading cause of death. That means there's probably few, if any, on this webinar tonight who have not been directly impacted by a neurological disease, either personally or because of a loved one, a family member, or a friend being affected by a brain disease or disorder. Which is why it's so important for an organization like the American Brain Foundation to fund research, again, across the spectrum of brain diseases, because we know that they're connected, so a discovery in one area can have a ripple effect on our understanding and treatment of others. And so that's why I'm really excited for the opportunity to speak with and introduce you to some of the American Brain Foundation's next generation research grant recipients who are working today to discover advances in treatments and the cures of tomorrow. Joining us tonight are members of the 2024 class to share more about their really exciting research projects and the impact that they hope to have on the lives of those affected by brain disease. After they share, we'll transition to a question and answer period where you can all ask our researchers your questions. And throughout the webinar, please feel free, as questions pop up, to put your questions in the question and answer. You just click Q&A at the bottom and just type your question in. And we'll get to as many as we can throughout the evening and at the end of when the speakers finish. I really hope you'll come away from this event tonight with hope that a future without brain disease is actually possible because of research projects like the ones you're going to be learning about tonight. But first, let me just tell you briefly about the American Brain Foundation's Next Generation Research Grants Program. Through these grants, we fund and support young, innovative investigators who are really the best and the brightest early career clinician scientists in the country. Over the years, the program has provided over $42 million to almost 300 researchers, most of whom have gone on to secure additional funding from the NIH and other national funders. In fact, almost 90%, 9 out of 10 who have been funded through these scholarships have gone on to receive federal funding. So as you'll see tonight, these grants are funding research, again, across the entire spectrum of brain diseases. And it really is our best hope to advance so that this is going to be translated into advances in treatments and cures. Definitely want to thank our funding partners for making this program possible, including a special thanks to our founder and research partner, the American Academy of Neurology. American Brain Foundation was started by the American Academy of Neurology, and we remain strategically locked together. And we raise funding to support the Academy's research priorities. Supporting these early career investigators is important, and I know because I was one of them quite a number of years ago, but it really was the first research grant that I received, and it really is what was responsible for kickstarting my career. So it's very gratifying to me to be able to sit here tonight and having come full circle and being able to recognize these young career investigators, because I know how important these grants are for the careers of these young investigators. And it really has been profound, actually, as chair of the board of ABF over the past 5 years to see the amazing work that we've been able to fund and some of how that's been translated, and how that's impacted the lives of people already. So this program truly exemplifies the American Brain Foundation's mission of bringing donors and researchers together to cure brain disease. So without further ado, let's meet our researchers. And let's start by hearing from Eleanor Thomas, Dr. Thomas from Emory University, and she'll be sharing a bit about her research into amyotrophic lateral sclerosis, or ALS, specifically looking at the blood-brain barrier as well as identification of biomarkers. So Dr. Thomas, please take it away.
Thank you, Dr. Dodick. Thank you for that introduction, and thank you to the American Brain Foundation and the AAN for your support of my project, and also the careers of young investigators. So what I've been doing at Emory is I've been using proteomic methods to study neuroinflammation in ALS. So as part of a large autopsy program, we're able to use proteomic methods to study ALS patient tissues. And we also have a biorepository where we're able to study plasma and CSF. And by integrating studies in these different tissues, we hope to identify immunological pathways that are dysregulated in ALS. The goal of this project is to find novel targets in ALS patient brain and plasma for future drug discovery, as well as to identify biomarkers that will be useful in clinical trials and biomarkers that help characterize early disease states and persons that carry ALS causing mutations that don't have the disease yet. So the study that really formed the basis for this project, we were profiling motor cortex from a set of sporadic ALS patients that don't have a genetic cause for their disease, as well as familial ALS patients with C9 mutations. And the main difference between controls and ALS patients in this study was a difference in various immune targets. We saw upregulation of IgM antibodies. So we're trying to define is what IgM is interacting with in the ALS patient brain, define antigens for IgM, as that will help us understand what the immune response is recognizing. We also hope to look at peripheral tissues such as blood and CSF to get a broader view about how the immune system is different in ALS versus controls by looking at different proteins in ALS plasma, as well as profiling lymphocyte populations in ALS blood. We hope to get a more complete picture for differences between ALS and controls. So my hope is that this research will lead to a better understanding of what drives motor neuron degeneration in ALS patients, and that by using human tissues, we're able to identify targets that will translate into therapies better for patients. And my other hope for this project is that we'll develop tools that will be useful in clinical trials for measuring treatment response and target engagement, in particular, tools for looking at biomarkers that target neuroinflammation or decreasing neuroinflammation in ALS.
Dr. Thomas, thank you. Fantastic. And particularly relevant since neuroinflammation is responsible, at least in part, for quite a number of neurological diseases. So when I talked about biological overlap and having a ripple effect, if you can understand neuroinflammation better in ALS, that can have a ripple effect on the understanding of other diseases for which neuroinflammation plays a role. So thank you for that. Now let's hear from Dr. Brittany Kurzanowski from the Barrow Neurological Institute. Her research explores air pollution as a major risk factor for Parkinson's disease. So Dr. Kurzanowski, take it away.
Thanks. Well, since there's no treatment to cure Parkinson's disease, prevention is absolutely key. And one way to prevent Parkinson's is by identifying the specific environmental toxins that are contributing to the disease, and then encouraging policymakers to eliminate or reduce these specific toxic exposures. But in order to do that, we need to understand the specific types of toxins at play. So earlier this year, we published a paper which identified a nationwide association between air pollution in the form of fine particulate matter and Parkinson's disease risk. And this work was notable because not only did we identify the association, but we also found a clear difference in the strength of the association by region. And this inspired us to consider why air pollution in one region might be a stronger predictor of disease compared to air pollution in another region. And we speculate that it might have to do with the different types of air pollution in these regions. So air pollution varies by source, and the sources differ by region. Air pollution can be made up of things like heavy metals or combustion particles or organic matter. And these things all have differing effects on the brain. So this work will help us, uncover the types of particles that are driving this relationship between air pollution and Parkinson's disease. I recently collaborated with the Mayo Clinic in Rochester, and we found evidence implicating traffic pollution in particular. And we actually just published this work in JAMA this past Monday. So if you're interested, you can have a look. In the study, we found evidence suggesting that not only does air pollution increase risk of Parkinson's disease, but it also increased risk of the more severe PD symptoms like dyskinesia. So what I'm working on now is, exploring a type of traffic component, elemental carbon particles. So elemental carbons, such as black carbon and organic carbon, those are types of traffic pollutants associated with diesel engines. And, these have been previously shown to compromise the blood-brain barrier and contribute to neurodegeneration. And the studies of elemental carbon have been relatively limited. So this work will, fill a knowledge gap and hopefully, identify a specific component in air pollution for us to focus environmental remediation, those efforts to reduce these kinds of toxins. So ultimately, we hope that we can identify a modifiable risk factor for Parkinson's disease that we can reduce and then therefore reduce the incidence of disease and the general burden of the disease in the nation. So thank you.
Excellent. Thank you, Dr. Kurzanowski. Really important work. Dr. Gazia Orniz, who comes to us from Massachusetts General Hospital. Can you tell us more about your research, which I believe is looking at post-stroke cognitive impairment?
Thank you. So thank you to the American Brain Foundation, the American Academy of Neurology, and the American Heart Association for this opportunity. So I'm a stroke neurologist, and I work in stroke, which, it's a very prevalent disease in the U.S. Every 40 seconds, there's a patient that develops a stroke, an adult patient. And by 2030, there will be over 3 million of adult patients, which is roughly 4% of the adult population. So currently, a patient after a stroke quite commonly can develop memory decline, which is known as post-stroke cognitive impairment. And we know that that can happen. The prevalence varies between 20%, but it can happen after 60%. So it's quite common. So my research focuses on finding clues, which are biomarkers that we find through a technique that's called metabolomics to try to understand how the disease works. And these clues, which are called metabolites, are basically the interaction of the patient's biology, the patient's genes, the environment, including diet and exercise. So our previous studies have found specific metabolites are associated with higher risk of stroke, but many of them were associated only to specific bacteria in the gut that can only be found there. So we hypothesized that this exact same gut-brain axis metabolites can be associated to developing cognitive decline in the future. Now, the way that we obtain these metabolites, it's by doing this technique, which currently this specific type of gut-brain axis metabolites are difficult to extract. So this grant would allow us to use a new method that we have developed in our lab to try to specifically look for this type of diet-related or environmentally related, and then eventually gut-brain axis-related metabolites. Our hope is eventually to bioengineer a therapy to try to target this specific type of gut bacteria.
Fascinating. I'm sure there's going to be lots of questions about this. Thank you, Dr. Garcia Juanes. Now let's turn it over to Dr. Hope Yang from Stanford University, whose research is evaluating cognitive decline and age-related memory impairment. So Dr. Yang, the floor is yours.
Thank you, Dr. Dodick, for the introduction, and thanks to the American Brain Foundation and its partners for funding my research. With aging often comes with lots of changes in our cognitive abilities, and memory decline is one of the most common complaints. However, the extent of memory decline varies widely among individuals, and the sources of this variance remain poorly understood. This is perhaps in part due to the absence of computational accounts of behavior, along with the use of rather coarse behavior assessments. So in our study, we will utilize a continuous and quantitative behavior assay of memory precision, namely remembering the perceptual detail about a past event. Combining this with computational characterization of the behavior, this approach has the promise to offer increased sensitivity to detect, to quantify, and to understand age-related memory changes, as well as its variability across individuals. The computational model is informed by recent data in younger adults, showing that memory precision is explained, at least in part, by variance in perceptual abilities. So a common example of this would be if you cannot really distinguish two different colors so well, you wouldn't be able to remember them too well. This naturally leads to the question whether and to what extent age-related difference in memory precision can be explained by perceptual ability differences, which also tend to occur with age. Another potential contributor to inter-individual differences in older adults' memory performance is amyloid beta burden. The presence of elevated A beta together with tau is linked to increased risk of developing Alzheimer's disease. And A beta burden is observed well before cognitive changes meet diagnostic criteria, and a significant proportion of healthy older adults demonstrate elevated A beta, perhaps reflecting preclinical AD. Therefore, the inter-individual differences in memory among clinically unimpaired older adults may relate in part to variability in their A beta burden. So we will leverage participants recruited from a large study, the Attention, Memory, and Aging study at Stanford, which is ongoing and include a large sample of both young and cognitively unimpaired, deeply phenotyped older adults with amyloid PET data obtained from them. So with this very rich data set, we hope we will be able to rigorously model age-related difference in memory as a function of visual perception and A beta burden, which will offer new insights into the mechanisms underlying subtle memory changes that come with healthy aging and potentially preclinical AD. Thank you.
Thank you, Dr. Yang. Thank you so much. Fascinating. Okay, our next researcher is from the Children's Hospital of Philadelphia, and their research focuses on variants in a certain gene associated with a spectrum of neurodevelopmental disorders, including epilepsy. Dr. Gillian McKee, look forward to hearing more about your research.
Thank you, Dr. Dodick, for the introduction. And I'd also like to thank the American Brain Foundation and the other sponsors of this award, the American Academy of Neurology, American Epilepsy Society, and the Epilepsy Foundation, for granting me this Susan Spencer Award. I'm a pediatric neurologist. I specialize in epilepsy and neurogenetics at the Children's Hospital of Philadelphia. And this funding will support my research studying the natural history of SYNGAP1, which is a rare genetic epilepsy, as well as identifying biomarkers and reliable outcome measures for disease progression and the response to treatment. Collectively, rare genetic conditions explain a significant proportion of neurodevelopmental disorders and epilepsy. However, given the variability within these conditions and the relatively small populations, it has historically been difficult to get a clear picture of the presentation of different symptoms over time. Excitingly, there are currently multiple different precision medicine therapies under development for many of these conditions, including SYNGAP1, but we lack the well-defined outcome measures critical to designing clinical trials and assessing the efficacy of these novel therapies. So SYNGAP1 encodes a key regulatory protein at the postsynaptic density at the interface of NMDA receptors and the downstream signaling apparatus in the cell, which is essential for synaptic plasticity. Disease-causing variants in SYNGAP are among the most common monogenic etiologies for generalized epilepsy and non-syndromic intellectual disability, with an estimated prevalence of about 6 per 100,000. And since its first description in human epilepsy in 2009, SYNGAP is now recognized as a common cause of developmental and epileptic encephalopathies, or DEEs, in addition to epilepsy with myoclonic atonic seizures and epilepsy with eyelid myoclonia. However, the recognizability of phenotypic signatures and the longitudinal disease trajectories in individuals with SYNGAP1-related disorders have not been clearly defined. So for this project, I'll use large-scale electronic medical record data integrated across various healthcare resources, including insurance claims data, medical records, and patient registries, and combine this data with that from an ongoing prospective natural history study to understand disease histories, define outcome measures, and recognize clinical subgroups to facilitate clinical trial readiness. From this retrospective claims data, I've already demonstrated the significant enrichment of behavioral features such as autism and aggressive behavior when compared to individuals with epilepsy as a whole, and more specifically when compared to those with Rett and Angelman syndrome, two similar neurogenetic conditions. I've also been able to extract medication data from the electronic medical records, and by analyzing that along with seizure frequency data, have been able to determine the comparative effectiveness of different anti-seizure medications. In terms of prospective data, to date, I've already evaluated over 80 individuals with SYNGAP in our research clinic within the newly established Center for Epilepsy and Neurodevelopmental Disorders, or ENDD, at CHOP and Penn. We're performing detailed assessments, including neurologic exams, standardized clinical scales, and reconstructing epilepsy histories, as well as collecting caregiver-reported outcomes. Additionally, I'll be using quantitative EEG to look for electrophysiologic biomarkers correlating with disease severity and progression. My work is a notable example of American Brain Foundation's Cure One, Cure Many motto in action. By developing a framework for integrating diverse retrospective datasets, we can delineate the natural history of various rare disorders, speeding up clinical trial readiness. While prospective natural history studies are the gold standard for validating outcome measures and generating control arm data for future clinical trials, they're often challenged due to recruitment difficulties, especially in these rare disorders. So using all available retrospective data prior to designing a prospective study allows us to focus the assessments on the most impactful outcomes and ultimately speeds up clinical trial readiness. So this funding will protect my time and support my ongoing training, allowing me to build a research program combining large-scale data and biomarker development to facilitate therapies for rare genetic epilepsies. Thank you.
Thank you, Dr. McKee. Really a great study, and amazing how you were able to combine data from so many disparate data sets. Very impressive. Next, we'll hear from Dr. Andrew Breithaupt from Emory University. Dr. Breithaupt will share with us his research into the possibilities of improving dementia diagnosis and how his research connects also the ABF Cure One, Cure Many motto. Dr. Breithaupt.
Thank you, Dr. Dodick. And I wanted to thank the American Brain Foundation, the Alzheimer's Association, and the AAN for the generous support. It's really been pivotal in starting my research career as a cognitive neurologist and care delivery innovation. Focused on widening access to evidence-based care for Alzheimer's disease and related dementias. So the project I'll be starting on is a first step in exploring how new generative AI based on large language models can help us detect, diagnose, and manage neurodegenerative diseases, starting with the detection and diagnosis piece. In neurology, the history is often crucial to diagnosis, and this is especially true for behavioral neurology. This, along with the physical exam, is really what we're often getting at when we say that we're trying to treat the patient in front of us. However, time and knowledge constraints have become large barriers to gathering this history, particularly for neurology, where the history can be long and complex. And there's increasing pressure to see more patients in less time, and the rapid expansion of medical knowledge exceeds what any one provider can know, and therefore know to ask every patient. And for neurodegenerative diseases, these challenges are urgent. As our population ages, dementia cases are increasing, but more than 50% are not diagnosed until it's too late for new treatments and many new promising clinical trials. And so our current state is that we're missing out on providing the majority of these patients with optimal medical care. And the majority of these patients never had a chance and opportunity to contribute to clinical trials if they had wanted to, clinical trials that aim to cure their disease. Generative AI is a new scalable technology that's been advancing very rapidly and may be able to help collect this comprehensive history with its ability to fluidly engage with patients, dutifully ask all recommended questions without any time limitations, and summarize lengthy text into a logical manner. And more recently, it's also shown to have really impressive medical knowledge with intelligent differential diagnoses. So for this project, we'll first just be using a voice-based chatbot that's powered by AI that will use a comprehensive questionnaire developed by a panel of dementia experts. And this is a questionnaire that mentor of mine evaluated and has been well studied in the primary care and specialty setting, paper form of this questionnaire at least. And then we'll be using AI to summarize what will likely be a lengthy interview into a history of present illness that a provider would be able to review before the office visit. We'll primarily be comparing this history created by AI to the history collected by a dementia specialist and then later in primary care. But we'll also be evaluating patients' experience with this modality, as well as exploring AI's ability to create an accurate diagnosis and management plan based on this history gathered. So the goal of this first project is to accelerate early detection and diagnosis for neurodegenerative diseases using generative AI, expediting access to quality care, emerging treatments, and clinical trials, and really trying to give every patient the chance to get that diagnosis as early as possible. One of the aspects about this project that really excited me from the beginning is getting at this American Brain Foundation's concept of cure one, cure many. Early detection and diagnosis, as well as time limitations, are huge problems across neurologic subspecialties. The demand for neurologists far outweighs the supply. And so my hope is that if this is successful, it can provide a roadmap so that every patient with any kind of neurologic problem can receive a diagnosis as early as possible. Thank you.
Crucial. Thank you very much, Dr. Brightest. Now we'll move to Dr. Jill Goslinga from the University of California at San Francisco, whose research explores how air pollution can affect ALS progression. Dr. Goslinga.
Thank you very much. I'm grateful for this chance to present my research focus, and I deeply appreciate the generous funding from the ALS Association and the American Brain Foundation that will help me accelerate this important work. As Dr. Thomas previously noted, ALS is a devastating neurodegenerative disorder that progressively destroys motor neurons in the brain and spinal cord, leading to loss of muscle function, respiratory failure, and death. And while often considered rare, ALS actually affects approximately 1 in 400 people, underscoring the significant impact this condition has on our population. As wildfire smoke exposure becomes more intense, frequent, and widespread due to climate change, studying its impact on ALS patients is crucial to identify vulnerable groups and potentially intervene to prevent respiratory failure, the leading cause of death in ALS. Existing evidence already suggests that air pollution exposure increases the risk of developing ALS, making it even more urgent to understand how wildfire smoke, which is a complex mixture of pollution, affects those living with this disease. Our preliminary study examined air pollution exposure among UCSF ALS patients from 2020 to 2022. And in 2020, a severe wildfire year, over 70% of our patients lived in areas with dangerous particulate matter thresholds, a measure of air pollution. This decreased to 40% in 2021 and 28% in 2022, which was still a high number despite it being a relatively light wildfire year. Our research is particularly important for uncovering health disparities, as certain populations may be disproportionately affected by wildfire smoke exposure due to geography, socioeconomic factors, other factors making it harder to protect yourself against smoke. For example, many major cities in the Western United States, like San Francisco, have a severe housing crisis. And so many people and developers are moving more inland to where there's more affordable housing. But this is pushing new construction into areas that are in the wildfire urban interface and regions with higher wildfire risk, increasing the number of people and properties vulnerable to wildfire and wildfire smoke. Insights gained from studying ALS patients could provide valuable information for managing other neuromuscular conditions as well, and help healthcare providers better protect at-risk patients across various diagnoses, as wildfires increasingly affect new geographic regions. By prioritizing this research, we can develop targeted interventions and bolster policies to mitigate the impact of wildfire smoke on vulnerable populations, potentially improving outcomes and quality of life for those with ALS and other disorders.
Thank you very much, Dr. Goslinga. Next, we'll hear from Dr. Francesco Michelasi from Columbia University, who will talk to us about his research on the potential treatments for peripheral neuropathy.
Thank you very much, Dr. Dodick. And I'd like to thank the American Brain Foundation, the American Academy of Neurology, and the Foundation for Peripheral Neuropathy. So about 50% of all patients receiving chemotherapy develop peripheral neuropathy, which manifests as pain and numbness in the hands and feet. The chemotherapy paclitaxel is one of the most closely associated with peripheral neuropathy. Unfortunately, the mechanism by which paclitaxel causes peripheral neuropathy is not well understood. And even more so, it's not even clear where the damage first occurs from paclitaxel. Interestingly, our lab has recently found that if you grow sensory neurons in a dish and you give paclitaxel only to the cell bodies, no degeneration occurs. However, if you give paclitaxel to the distal neurites, which is about the equivalent of where the sensory nerves come to the skin, you get degeneration of those neurites. And so also interestingly, right before this degeneration occurs, in the distal neurites, there's a depletion of the mitochondria, which are the powerhouses of the cell. And if you give a drug that boosts mitochondrial biogenesis, which this one is called ICAR, the degeneration caused by paclitaxel is prevented. So as such, I'm looking to three questions. One, does mitochondrial injury at the distal nerve terminal lead to degeneration, or is it just an association? Two, what specific pathway in the mitochondrial biogenesis prevents paclitaxel-induced peripheral neuropathy? And three, can ICAR actually prevent paclitaxel-induced peripheral neuropathy in a mouse model? And I hope that through these efforts, we can get closer to developing a treatment for chemotherapy-induced peripheral neuropathy. Thank you.
Thank you very much, Dr. Michelasi. Very clear, crystal clear. Thank you, and important. Finally, let's hear from Dr. George Kannercott about his research into where misfolded proteins called alpha-synuclein originate in the body and how they might contribute to neurodegeneration in Lewy body disease.
Thank you for the introduction, Dr. Dodick, and thank you to the AAN, the American Brain Foundation, and the Alzheimer's Association for your research support. So I'm a Parkinson's disease doctor at the University of Pennsylvania, and like many brain diseases, Parkinson's disease is characterized by misfolding of proteins, which are the building blocks of cell machinery. And we're increasingly recognizing that some of these misfolded proteins are found not only in the brain, but outside the brain. And so my research is focused on one of these forms of alpha-synuclein that's outside the brain, which we can use to identify blood from people with Parkinson's disease and differentiate them from those that have related conditions like dementia with Lewy bodies or people that don't have Parkinson's disease. And interestingly, these specific misfolded forms of alpha-synuclein are associated with different rates of cognitive decline in these individuals. And we identify these specific misfolded proteins in extracellular vesicles, which are basically packets of information that are circulating in the blood that cells use to communicate with one another. And so the main focus of this project that's been funded by the American Brain Foundation is to identify where these misfolded forms of alpha-synuclein are coming from, whether it be from the brain or outside of the brain. And then to figure out how do those misfolded forms of alpha-synuclein outside the brain relate to causing the spread of pathology that we see within the brain. So hope to answer some of the fundamental questions for how Parkinson's disease and related conditions start. So where does it start? How does it start? And ultimately, how does that pathology spread from one area of the body to the other?
Fantastic. Well, what a lineup and what a collection of research projects. Really, really impressive. Now, everyone, we have a unique opportunity here for all of you to begin to ask some questions of our next generation research scholars here. So please, if you have questions, please type them into the Q&A or the chat, and I will pick them up and ask our panel. So we have a question here. I don't know, this is not a question about anything necessarily that any of you presented, but the question is a traumatic brain injury considered a brain disease? Does anybody want to tackle that?
I'm happy to help start to answer that. So traumatic brain injury is direct injury to the brain. It can result in life changing impacts and long lasting impacts, even if it's not recognized at the time of the initial injury. And we know that it puts people at risk for future brain diseases, including Parkinson's disease, Alzheimer's disease, even linked to ALS and many others. So I think yes, it is. It is not only a disease entity in itself that we're trying to, as a neurology community, better understand and classify, but also understand how it puts people at risk for other more well-defined brain diseases later on in life.
Yeah, I would say certainly traumatic brain injury. There's different degrees of traumatic brain injury, and the most common one by far is mild traumatic brain injury, otherwise known as concussion. And I'm sure many people on this panel here and many in the audience have probably had a concussion in their lifetime. It's a very common thing. I wouldn't necessarily consider that a disease, but as Dr. Kanterkot mentioned, traumatic brain injury is an independent risk factor for cognitive decline and other neurological diseases. And certainly repetitive traumatic brain injury or repetitive concussions, as many of you have become aware of in especially in collision and contact sport, can give rise to a disease called chronic traumatic encephalopathy, which is a progressive neurodegenerative disease. So I don't want anybody out there thinking that a single or even a couple or even a few traumatic brain injuries or concussions, you have a neurological disease or you're at risk for a progressive neurological or neurodegenerative disease. But certainly multiple repetitive concussive head impacts and even subconcussive impacts where people hit their head repeatedly but don't necessarily experience symptoms can give rise in uncommon instances to a progressive neurodegenerative disease. Okay. Is frontotemporal dementia, Alzheimer's disease, Lewy body disease related to ALS? I've heard many people state in their family of members who had both. So a number of you spoke on ALS or Alzheimer's or Lewy body dementia. So I'm just going to throw that out there to whoever would like to take it and encourage the others to pile on.
I can start. Well, there's a clear association between frontotemporal dementia and ALS, and those are both linked to TDP-43 pathology, which is a shared co-pathology between the two disorders. So we find TDP-43 aggregates in patients, autopsy tissues from patients with FTD and ALS. In terms of, I think there's another question in there about Lewy body dementia and other associated dementias that have different pathologies. So Lewy body dementia is usually an alpha-synucleinopathy, which is a different misfolded protein. However, there is a lot of research looking at how, what we see at the autopsy level in patients at advanced ages is they don't always have one pathology. They can have shared pathologies. So trying to really figure out to what extent symptoms are related to alpha-synuclein aggregates versus TDP-43 aggregation, or even some ALS patients may have a little bit of amyloid pathology. To what extent one of those is contributing to dementia is an active area of research and an interesting question.
Anybody else would like to comment on that? Okay, maybe I'll turn to and ask Dr. Kurzanowski a question. So you talked about air pollution and its link to Parkinson's disease. In fact, correct me if I'm wrong, but Parkinson's disease is the fastest growing neurological disease on planet Earth. And it's thought that that is probably related to environmental toxins and particulate air pollution, et cetera. So you're studying elemental carbon specifically. Is it true that there are areas in the United States and maybe more broadly internationally where the prevalence of Parkinson's disease is very low in regions where elemental carbon and particulate pollution is low? And correspondingly, or in contrast, are there areas in this country and in other countries where elemental carbon is very high and there's a corresponding increased prevalence of Parkinson's disease? So in other words, do the two go together in various regions that are either very high or very low in elemental carbon?
That's a really good question, and I can talk about it in terms of my prior study, which looked at particulate matter. Actually, if I'm allowed to share slides, I would show some pictures, but otherwise I don't have to. Would you like to see?
Sure, I would. Yeah, absolutely.
All right. I'm going to go ahead and share my screen. Okay, so if we were looking at my study from last year, we can see that the hotspots for Parkinson's disease were here in the kind of the Mississippi, Ohio River Valley, while the cold spots were here in the West. However, one of the interesting features of that study was the association, and I kind of got at this a little bit where I was saying that we noticed that different regions had stronger associations. So the association was easier to detect in this area right here. So, which is kind of particular, right? Because if you remember the last slide, that's a cold spot. So there's low prevalence or low incidence of Parkinson's disease. There's also low levels of PM2.5 over there. So what's curious then is why is that? And what you may have already noticed from this plot that I put next to it is actually it's just a factor of the association. It's a factor of the trends we're seeing in the scatter plot here. So these counties over here correspond to the place in the scatter plot that has the steepest slope, which is the strongest association. And so really this type of association map is just showing, it's an artifact of the ceiling effect. And so there's been maybe, I don't know, maybe 6, 7, 8, 9, 10 studies that have identified this plateau effect in PM2.5. And so the idea here is it's possible it's due to a natural biological threshold. So perhaps PM2.5 can only, cause its cellular degradation up to a point. But the other theory is it has to do with the types of air pollution, right? Because if I'm just modeling PM2.5, that's not, that is a size of a particle. That's not a type of a particle. And we already know that dust is going to affect you differently than diesel particulates and things like that. And so we find that that plateau effect in other studies as well. The one with Mayo Clinic also. But in terms of elemental carbon, you can think of using data on traffic pollution. Also, we obviously have the elemental carbon estimates themselves to look at. But these, this is, I could talk more, but there's a lot of things going on. So you can think about how traffic pollution interacts with, VOCs from trees and things like that. So there's a lot of things that we need to think about when we're thinking about air pollution, and it's a very complex problem. And there's also the comment that was placed in the Q&A is multi-diseases, right? So some of these studies that are coming out, we have Medicare data or maybe a large hospital data set. You can look at Parkinson's, ALS, you can look at Alzheimer's. And so there's just a lot to do, and there's a lot of opportunity for collaboration as well.
Thank you. Thank you, Dr. Krasnowski.
Yep.
There is a question here. Can Lewy bodies be found at autopsy in people without a clinical diagnosis of Lewy body disease or Parkinson's disease, or even symptoms of any of those alpha-synucleinopathies? I'm paraphrasing now a little bit. And so who wants to take that question?
So I can start. So yes, there's a certain percentage, probably around 5 to 8% of people at autopsy that have Lewy bodies that we don't they don't never had any evidence of clinical Parkinson's disease or any kind of memory impairment. And it's not clear that, if they live long enough, would they manifest symptoms of that disease? And also, even with some of the now not newer clinical biomarkers, so you might have heard about the skin testing and the spinal fluid testing for misfolded alpha-synuclein. That same rate of positivity in people that don't have disease is also there somewhere in kind of the 5 to 8% range. And so it's not clear whether that puts people at increased risk. We think it probably does. But we're still trying to figure out what exactly that means to have incidental Lewy body disease or even incidentally misfolded alpha-synuclein. What does that mean for your future?
Yeah, yeah. And of course, we know, like many of these neurodegenerative diseases, the pathology starts being laid down years to decades before the onset of symptoms. So, yeah. I love that we are looking at causes and have identified air pollution, fire, traffic, etc. What about food and clothing? So I guess the question here is really about other types of environmental and organic toxins that have been linked to Parkinson's disease and perhaps other neurodegenerative diseases. Anybody have a comment on that particular question?
I have a little comment. So we've also been looking at pesticides at Barrow Neurological, and it's very difficult to use our data set to investigate the food system because we can't identify using population data who's eating organic and who's eating non-organic. One thing you could consider is water, like the water coming out of your tap. Is it brown water or is it surface water? Is it from a shallow aquifer? Is it from a deep aquifer? There's another thing I was looking at recently, but I can't remember. The municipal wells, things like that. But you can try to figure out ways to gather data sets that could get at the question. But it's very difficult with the food. In particular, when you think about where pesticides are applied to certain crops, those agricultural crops are usually shipped and sold to other communities. There is data that would track where those go, and so that there's opportunities, but we haven't got there yet.
Great, thank you. Let me go to Dr. Brightcott. You talked about large language models and generative AI and detecting and making an earlier diagnosis, of course, and we understand why that's crucial because there's often a delay in diagnosis because, as you say, that we can't meet the demand, the capacity can't meet the demand. I'm just a little bit interested in this voice bot-based chatbot. So is the idea that the patient will, with an examiner or a doctor, undergo a lengthy interview, as you say, and then that's AI is used to interpret that history that's taken and apply a diagnosis? Is that the idea here?
I think that's getting at the eventual idea. So the part that I'm testing out now kind of came about from being at an institute where we would see a lot of patients in clinic, and we'd refer these patients to a research setting where we would, another cognitive neurologist would need to verify everything and go over the history themselves. And what I found is that history that it took my colleague an hour and a half to go over I could verify with the patient in about 15 minutes. And that sometimes I would find something that was a little bit off, and there might have been a misunderstanding, and oftentimes it wasn't clinically meaningful. But that's kind of the birth of this project, was thinking that we can really we can still verify this. So even if these large language models are imperfect at times, that we can still verify what they collect in a very short amount of time. And then during these appointments, it was great because I had more time to build rapport with the patient, dig in a little bit deeper, and really felt like I could come into the appointment already having a very good idea of what was going on. So I felt that alone helped me to come to a diagnosis sooner and quicker and more accurately. And so that's the part that we're really honing in on this project. But I can't help but test out these large language models, especially a secure instance of GPT-4, given its really impressive abilities with when I plug in histories. I am astounded sometimes at the differential diagnosis that it comes up with that is, on par with a lot of experts. It's not perfect. That's not the case with everything, but that's my hope, is that there would be a clean pipeline where even before coming to the appointment, patients would be able to engage with this kind of chatbot and provide the history that's needed for the provider to give them that opportunity for an early diagnosis.
One can imagine a future application of this, which is where as cognitive health assessments become eventually more common in neurological practice, despite the presentation, or even in primary care, you'll be able to detect, people with Alzheimer's disease pathology, even in the absence of symptoms. Preclinical,
yes.
Right?
Yes. Yeah.
Yeah.
That's the hope.
That's the hope. So Dr. Canercott, you talked about the potential of using whatever you're looking for in the blood, alpha-synuclein aggregates. I don't know whether you're studying exosomes, or I'm interested to know what exactly you're studying, and how confident are you that the alpha-synuclein, are there different isoforms of alpha-synuclein or types of alpha-synuclein that will allow you to differentiate Parkinson's disease from Lewy body disease from other alpha-synucleinopathies? And by the way, just a question for the audience, if they're not coming from the brain, this is a provocative question, where could they be coming from?
Yeah, no, these are all great questions. So what we're using is we have actually antibodies that can recognize different shapes or folds that alpha-synuclein takes on. And alpha-synuclein is notorious for taking on different shapes and actually coming together as different multimers or oligomers. So we have antibodies that we can use to differentiate these different forms. And I think the best evidence now is for using those forms to differentiate multiple systems atrophy from Parkinson's disease and dementia with Lewy bodies. We have antibodies that can differentiate dementia with Lewy bodies from Parkinson's disease just using blood. And so the specific forms that we can identify are with these antibodies are also present in exosomes or extracellular vesicles. And each of those vesicles are kind of packets that have their own unique nucleic acids, proteins within them as an address for where their cell of origin was from. And so hoping to answer the question, by pulling out these specific forms of synuclein in these extracellular vesicles to figure out where their origin is from. And it could still be from the brain, but this is an unbiased way to answer that question. Because increasingly we're seeing these forms of synuclein or abnormal forms of synuclein identified outside the brain, the gut, the nasal mucosa, the skin, and we still don't understand why they're present there, and what is that the place where they're coming from or not.
Very good, excellent. Dr. Michelesi, is there any observational data or hints in the clinical literature that any therapies that boost mitochondrial biogenesis or preserve mitochondrial function because of their antioxidant properties actually have a beneficial effect on either preventing or mitigating peripheral nerve injury from chemotherapy?
Yeah, that's an excellent question. So there is, I believe, a few studies that were done that showed that the antioxidant N-acetylcysteine in, I believe, oxaliplatin chemotherapy has been shown that it can help prevent at least development of the painful peripheral neuropathy. And there's been a lot of data out there about sort of that, a lot of the problem is that a lot of these chemotherapies do a lot of different things. And so, one, they can do their mechanisms of action, and then they all have 50 other things. And so one of them is mitochondrial damage. So the mitochondria tend to take a big beating from all of these. So these are sort of the observational things that would suggest that maybe treating the mitochondria will be helpful.
Great, thank you. There's a question in the chat about any research on the incidence of parietal lobe epilepsy or sensory seizure in children and or adults. Can anybody answer that question?
I can take a stab at it. It's definitely a less frequent form of epilepsy. But especially in adults, temporal lobe epilepsies tend to be more prevalent. But it's in children, at least, and that's my bias as a pediatric epileptologist, seizures can come from anywhere. But I don't have exact numbers for you. I'm sure they are out there, though.
Thank you. Dr. Yang, is there any evidence that amyloid pathology in the absence of cognitive symptoms, could that give rise to these perceptual differences that you're talking about that may precede the more characteristic features of Alzheimer's disease?
Yeah, there's some evidence suggesting that this type of preclinical biomarker burden may lead to some changes in the visual system, like macular degeneration. But I think that is rather atypical for AD. And that's actually not the targeting cohort for my study, because we are going to screen their visual acuity and normal color perception before we enroll them in the study. What we are targeting is more of what we call cortical perceptual deficit. If they have some vision impairment or changes in color perception due to cortical mechanisms rather than anything happened earlier than that.
See. Thank you. And Dr. Garcia Juaniz, you're looking at gut-brain axis metabolites and whether that's predictive, I suppose, of post-stroke cognitive impairment. Could the gut-brain axis dysfunction or dysbiosis actually increase the risk of stroke? I guess that's my first question. And the second question, how are you going to parse out whether these independently increase the risk of post-stroke cognitive impairment versus the location of the stroke? Because we know that depending on where the stroke is, that can have profound effects on cognition. So how are you going to separate that?
Yes, those are excellent questions. I think the first one about the gut-brain axis metabolites specifically. So the specific sample that we have is for serum studies of patients on the regards cohort, which is specifically 50% African American and 50% white in a stroke buckle and a stroke belt. So really high risk individuals on the area. I think in terms of how to define if they are related to the gut-brain axis, it's basically knowing which one they are, and then you look for where they are produced, basically. And the ones that we did find, which were guanosine and pseudouridine, and then others like indole-3 propionic acid, come from E. coli. So they cannot come from otherwhere else in your body. So that's how we define that they are specifically related to some bacteria. I think that the question eventually is which bacteria and what bacteria in your microbiome specifically. And that comes from getting stool samples from patients, which unfortunately we don't have in the regards cohort, but this study will help us create data to potentially apply for future funding and then being able to correlate that and specifically show which bacteria they are. And the second question you had was where specifically the area of the stroke. So there is some limitation in our study, which is that it was done the cognitive data over the phone. So patients that are aphasic would not be, answering that well to the questions. So there's definitely going to be a limitation there. And I think again, future studies could be able to answer that.
Great, thank you so much. Well, unfortunately, it's at the top of the hour, and I'm sure you all have additional questions, I certainly have additional questions, but this has been really a great evening. Thank you so much to everyone for attending this evening and for your terrific questions. And to our researchers, thank you very much for giving up your time tonight and sharing your projects. Really, really impressive. And it's just exciting to see the level of sophistication of these studies that no doubt will have an impact and will be, the results will, as any good research, it will generate more questions. And I'm sure you all will be successful in securing additional funding, as most of our recipients have, to continue on in your research career. So thank you again. And, remember, anybody who's attending tonight, to, the support that you give to the American Brain Foundation supports this kind of research. And so it's really crucial to the mission of not only the American Brain Foundation, American Academy of Neurology, but to the future of neuroscience research in the United States and abroad. So thank you very much, everyone, for attending this evening. Have a good rest of your evening. Take good care, and we'll see you next time. Thank you.